Hello, dear students. Today we are going to start the third chapter in this unit, which is chemical reaction in living organism's body. We talk about the structure of the living organism's body. Now we are going to talk about the reactions happen inside the living organism's body. Living organism's body, their life depends on the occurrence of group of biochemical reactions. These reactions are called metabolism, or they are called metabolism. What is the meaning of metabolism? Group of biochemical reaction take place inside the living cells, in which the living molecules may be break down, or they are built from simple molecules forming complex one. So they are divided in two types of reaction. The first one is called anabolism. Anabolism, where complex and macromolecules build from simple one. This is type of metabolism. We are converting complex one to we are building complex one from simple molecules. The second type is catabolism, and catabolism, where the molecules are broken down to excrete energy. This is called catabolism. So the metabolism, or any reaction of the biochemical reactions, is called anabolism or catabolism. The first type, anabolism, process by which simple molecules are used to build the complex macromolecules through a group of chemical reaction which consuming energy. We use energy in this chain of reaction, so we are building complex substance or complex molecules, and this used in synthesis of protein from amino acid, photosynthesis process inside the green plants. All these are types or are examples of anabolism. The second type, catabolism. It's process of breaking down the chemical bonds between atoms molecules to extract chemical energy stored in them. We are going to take energy from this reaction. We are breaking. So we can break glucose to release energy from it. When cell respiration happens, they are breaking molecules, so the energy comes out from it, so it is called catabolism. The importance of metabolism for growth of body, or repairing damaged tissue as anabolism, or obtaining energy as catabolism. To continue this happen, or these reactions happen, we have something called enzymes, which help in the biochemical reactions or the metabolism reaction. They need energy. We use enzymes. What is enzymes? Enzymes are biological catalysts that are made up of protein molecules, work in increasing the speed of chemical reaction which happen inside the living organism. These enzymes are natural catalysts inside the body which speed up the chemical reaction. They decrease what is called activation energy. Any enzyme decreases the activation energy, as the metabolism or types of reactions need high activation energy. What is the meaning of activation energy? Minimum energy needed in order for chemical reaction to occur. Any reaction needs amount of energy to happen. So when we add enzyme, this amount of energy decreased, so the reaction will happen faster. So we need the catalyst to decrease the activation energy and speed up the chemical reaction. What are the properties of the enzymes? The enzymes are similar to the chemical catalyst. They resemble the chemical catalyst. They only speed up the chemical reaction without taking part of it. They participate inside the reaction and speed it without affecting or consuming. The enzymes never are consumed or affected by the reaction, only they speed it. They are affected by the concentration of the hydrogen atom and temperature, the pH value, and the temperature affect on the action of the enzyme. The enzyme differs from other catalysts. Every enzyme is specialized for only a special reactant. They can work on a special substance or special substrate. They are highly specific. Each enzyme is specific from one reactant or substance. These substrate or only work for a few types of reaction. The enzyme reduces the activation energy used for starting the reaction. So the reaction never interfere or take part inside the reaction. It is affected by both pH value and a temperature. They are specialized for special type of substrate, and they can do small number of reaction or maybe one reaction only. They decrease the activation energy needed for the reaction to occur. If we look here, this black part is the enzyme, and the substrate or the reactant which starting the reaction will join to the enzyme and form what is called enzyme substrate complex. Then the reaction happens and the product is formed, and it leaves the enzyme in the same shape without any change. So if we look here, the enzyme at the beginning and the enzyme at the end the same. And if we look here, we have what is called active site. The active site, the location of meeting of an enzyme with substrate. So each enzyme is specific for a special type of substrate because it has a special location which is called active site. And the substrate is the substance which enzyme work on, the reactant which start the reaction. It work by forming complex, then it leave the reaction without any change. This is how the enzyme work. Now we are going to talk about factors affecting the work of the enzyme. We said we have two factors, temperature and pH value. The temperature effect on the enzyme. Every enzyme has a special temperature or what is called optimal temperature. And enzymes are sensitive to thermal change. Why? Because any enzyme is formed of protein. These enzymes determine their activity in a narrow range of temperature. We need a special temperature for the enzyme to work because it is made of protein substance, and proteins are sensitive to temperature. So each enzyme has optimal temperature where it is more active, and these range from 37 to 40 Celsius. Enzyme optimal temperature where the temperature it is most active, and it ranges from 37 to 40. The enzyme activity gradually decreases when the temperature rises above the optimal temperature. Until it reaches certain temperature, it will stop completely. Why the temperature rises will stop the enzyme activity? Due to the change of its normal composition. The enzyme will not restore its activity even after decrease, because it will denaturate. It will not work again, stop working. The temperature, when it decreases below the optimal temperature, the activity decreases until it reaches minimum value, and it is low, and it will stop at 0 degrees. But the enzyme can restore or reactivate again by rising the temperature. So here, the increase of temperature more than the optimal temperature, it will decrease the activity until it will stop and never work again. It's called, it's denatured. When the temperature decreases than the optimal temperature, the enzyme will stop, especially at 0 Celsius. But when the temperature rises again, it will work again. Now we are going to talk about the second factor affecting the enzyme activity, which is the power of hydrogen, or the pH value, the concentration of hydrogen ion. The pH value is the measurement that determines the concentration of hydrogen ion in the solution, whether the solution acidic, basic, or neutral. We can call this solution acidic according to pH value less than 7, neutral when it is 7, and basic when, or alkaline when it's more than 7. So the value of power of hydrogen solution ranges from 0 to 14, depend on the positive hydrogen ion concentration. Enzyme affected by the changing of pH value because the protein substance contains acidic carboxyl group and basic amino group. Each enzyme has an optimal pH value which work with maximum efficiency. If the pH is lower or higher than the optimal enzyme activity, it will decrease. So the pH value is optimal, enzyme activity will be the maximum efficiency. But when the pH value increases or decreases, then the optimal pH of the enzyme, the activity of the enzyme will be affected. Enzymes work in different pH, like pepsin enzyme. It is one of the enzymes found inside the stomach, and it is work in pH acidic. So they range from 1.5 to 2.5, and this is the maximum activity for pepsin enzyme. We have other enzyme called trypsin, which inside the small intestine, and it is basic. It's range from 7.5 to 8. So these different values of pH value and different activity of different. Each enzyme has its own optimal temperature. If it increases, then the optimal, it will not work effective, or if it will decrease, then the optimal pH value, it will not. Most of the enzyme work at 7.4 pH value because its molecule contains acidic carboxyl group and amino basic group. But we have some exceptions like pepsin enzyme, which is found inside the stomach. It is work in acidic pH value 1.5 to 2.5. Other example is trypsin enzyme. Trypsin enzyme works at basic pH value, which is more than 7.5 to 8. This is the optimal pH value of the enzyme. Other factors affecting the enzyme action is the concentration of the enzyme, substrate concentration, and presence of inhibitors. Inhibitors are substances which stop the action of the enzyme. So we talk about the enzymes, their importance, as they reduce the activation energy, reduce the cell consumption of more energy, and ensure that the chemical reaction happen very quickly. At the end, the word enzyme is coming from Latin word which means yeast, and it is discovered during the glucose fermentation into alcohol by yeast. That's for today. Thank you and have a nice day.